Hello, my name is Joan Han. I'm a pediatric endocrinologist at Le Bonheur Children's Hospital and the University of Tennessee Health Science Center in Memphis. Today, I will be repeating a talk that I gave in October of 2018 in Ann Arbor, Michigan for the PCORI conference, with a few updates because it's now July of 2020. The title of my talk is How to Train Your Dragon, Families and Researchers Working Together to Improve Clinical Outcomes. These are my disclosures. I titled this talk because of the popular movies that my kids love, and the major theme of the story is how important it is to collaborate. That the trainer and the dragon together can accomplish great things because they work together. And I think that that's true also for researchers and families participating in research, that it's really that collaboration that allows for really important medical progress to happen. Today, I'll be sharing about my work at the National Institutes of Health, and go on to the next slide. When I was a fellow studying pediatric endocrinology, I had to choose a research project. And I decided that I wanted to study WAGR syndrome because even though it's a very rare condition, I was really interested in the potential for studying the rare to learn about the common and together be able to help children with both rare and common disorders through this approach. So WAGR syndrome occurs in about 1 in a million individuals. The WAGR is an acronym for its symptoms. Wilms tumor is a tumor of the kidney. A is for aniridia, which is absence of the iris. And you can see here, it's like in photographs, there's a lot of red eye appearance. G stands for genitourinary anomalies. This is a boy that has microphallus and undescended testes. And R stands for a range of developmental delays. The cause of WAGR syndrome is heterozygous 11p13 deletions. The size can be about 1 million base pairs to really large, like over 26 million base pairs. The core symptoms of the classical features are caused by loss of WT1, which is responsible for Wilms tumor and GU anomalies. Loss of PAX6 causes aniridia. The cause of the developmental delays, though, was not well known at the time. And then there are some patients who have larger deletions that affect EXT2, and that causes osteochondromas or exostoses of the bones. And then ALX4, if you have that deleted, then you can get holes in the skull. So these are parietal foramina that you can see in this X-ray. Some, but not all patients develop hyperphagia, which is excessive hunger and eating and subsequent obesity. And then some, but not all patients develop impaired nociception, which is insensitivity to pain. And the hypothesis that I had was that one of the genes in this region called Bdnf, brain-derived neurotrophic factor, is only about 4 million base pairs away from the WAGR critical region and is deleted in about half to a third of patients. And so the question is, perhaps maybe Bdnf is what's causing these extra symptoms. So what is Bdnf? It's a 27 kilodalton homodimeric protein that's widely expressed throughout the nervous system. It's important embryologically for nervous system development and postnatally for synaptic plasticity and neuronal survival. It's encoded by Bdnf at 11p14, and it spans 67 kilobases. That's a very big gene. There are 11 exons with 9 promoters. There are 5' untranslated exons that are variably spliced with the final 3' coding exon. And so all these different combinations allow for tissue and brain regions specific expression. In mice, we know that if you have Bdnf haploinsufficiency or loss of one copy of the gene, that they develop hyperphagia and obesity. They also have reduced sensitivity to pain, and they've impaired social interactions, learning, and memory. As you can see, this mouse here, he weighs twice as much as his wild-type litter mate. And so when you think about the symptoms that happen in the mice and the symptoms that can happen in people, we had the hypothesis that Bdnf haploinsufficiency is what's leading to the obesity subphenotype in WAGR syndrome, as well as the neurocognitive impairments that you can see in some patients. So the purpose of the research study was to characterize the metabolic and neurocognitive phenotype of Bdnf haploinsufficiency, and to also look at the phenotype-genotype correlation for other genes in this region. we recruited patients with WAGR 11p deletion syndrome and looked at their deletion boundaries by using oligoarray CGH. So that's comparative genomic hybridization and followed by sequencing to get the exact base pairs for the boundaries of the breakpoints. We also had patients serve as controls who had visual impairment, and those are patients who have isolated PAX6 mutations, so they only have the aniridia component, and also healthy controls who were age, sex, race, and BMI matched. Patients came for a comprehensive inpatient evaluation, so they had to stay for a whole week, and you can see this is the schedule in table format and then kind of more visually to the schedule. So I am incredibly grateful to the patients and their families for volunteering to do this. This is really a very gruesome schedule, actually grueling schedule. They, basically you can see it's packed with testing. So I'm incredibly grateful for the heroic sacrifice that the patients endured in order to make progress in medical science. So I'm incredibly grateful. So what did we learn from these studies? So let's start with Bdnf, because that was our main hypothesis. So Bdnf, the reason why we were interested in the first place is that it's believed to be an important part of the leptin signaling pathway. So adipocytes, which are the cells that make up fat tissue, make leptin, a hormone that travels in the bloodstream to the brain and acts within the hypothalamus of the brain to upregulate signals that tell you to decrease eating and to also suppress signals that tell you to eat more. And then also it upregulates signals to burn calories by increasing energy expenditure. So the net effect is catabolism, which would induce weight loss and prevent excessive weight gain. And brain-derived neurotrophic factor, based on animal studies, functions downstream of this leptin signaling pathway. So when we looked at patients with deletions that spared Bdnf, so those are the plus individuals, versus those who have deletions that include Bdnf, those are the plus individuals, you can see that those with intact Bdnf over toddlerhood to adulthood tend to have normal weight, and that those with Bdnf deletion tend to have obesity. And then when we looked at this quantitatively using body mass index Z-score, so that's the BMI adjusted for age and sex, you can see that the yellow bars, which is Bdnf loss, has higher BMI throughout childhood, and then compared to those with intact Bdnf. And then when we examined prevalence of childhood obesity by the time age 10 is reached, we observed that 100% of children with Bdnf deletion had obesity by the time they were 10 years old, compared to only 20% of those with intact Bdnf. And that's similar to the background prevalence of obesity in the United States. In addition to just looking at weight, we also looked at body composition using dual energy X-ray absorptiometry. That allows us to look at fat, muscle, and bone compartments separately. And we confirmed that it's indeed adiposity that is increased to explain the higher weight in patients with Bdnf loss. When we looked at the distribution of fat, whether it's subcutaneous or under the skin versus visceral, which is inside of the belly, we observed a similar visceral to subcutaneous fat ratio. And what this tells us is that patients with Bdnf loss have more fat, but how it's distributed is similar. There isn't abnormality in fat distribution. And we all know that visceral fat, or the fat that's inside of the abdomen, is what's at risk for metabolic syndrome. So patients with Bdnf loss do have higher rates of high cholesterol, hypertension, insulin resistance, and diabetes. But once you adjust for degree of adiposity, then all of these factors are similar in those with and without Bdnf loss. So that means that if one could control or prevent the obesity, then one could also prevent many of the metabolic complications of obesity, which is reassuring. When we think about what causes weight gain, it's an imbalance in calories in versus calories out. So what we looked at was intake using a hyperphagia score, and that's a questionnaire that looks at how much a child is thinking about food, driven to looking for food, including extreme measures like searching the trash and sneaking food, and how much it affects their day-to-day life. Are they thinking about food so much that they can't get schoolwork done or other concentration problems because they're focusing on food? And so patients with BDNF loss have a higher hyperphagia score compared to those with intact BDNF. And in fact, this score is actually quite similar to patients with Prader-Willi syndrome, which is another syndrome that is known for its hyperphagia component. On the other end of the energy balance is energy expenditure. So we used indirect calorimetry to look at the basal metabolic rate of patients. This is a plastic covering that allows for measurement of carbon dioxide production and oxygen consumption. So this kind of spacesuit kind of setup allows us to measure the rates of exchange of these gases. And so when we looked at this, we observed that resting energy expenditure is similar for those who have BDNF loss and intact BDNF. And this really kind of confirms the idea that it's hyperphagia or overeating rather than decreased energy expenditure and abnormal metabolic rate as the cause for obesity. And so in mice, we actually observed something similar in that if you pair feed the mice, and what that means is you restrict the amount of food that the mouse gets so that it's matched with what its wild type litter mate gets, then you can actually get them to be the same weight. You can prevent the obesity in the mice. And so I think that the fact that we observed that there wasn't a resting energy expenditure problem in humans is also reassuring that if we can prevent or at least ameliorate the hyperphagia component, we might be able to prevent or reverse the obesity. We were interested in making sure that there was not an additional locus besides BDNF that was important for obesity. So we lined up all the patients based on their centromeric deletion boundary. And here the color coding is normal weight, blue and yellow for obesity. And you can see that the pattern of those with obesity or normal weight, essentially, doesn't associate with the centromeric boundary, indicating that there was no particular critical region for childhood obesity on this end. On the other hand, if we line them up based on their telomeric boundary, you can see that actually based on the yellow and blue bars that a pattern does emerge. It turns out that every patient that has a deletion that encompasses where this red line is develops childhood obesity. On the other hand, everyone who is normal weight throughout childhood actually has a deletion that stops before it gets to this red line. And this red line happens to be BDNF. So that really does kind of confirm our initial hypothesis. And then when we zoom in on the boundary here, borders. So what's interesting is that this patient who has developed obesity has deletion of only the first 3 exons of BDNF. So these are the 5' untranslated regions sparing the single protein coding exon 9. So this seems to suggest that loss of just even the 5' UTR is enough to cause obesity in WAGR syndrome. On the other hand, this patient who was normal weight had a deletion that ended about 72 kilobases upstream of where BDNF begins, and there are no protein coding regions in this interval. Again, confirming that really BDNF is the candidate gene for causing obesity. So given the fact that we had observed obesity in someone with only exons 1, 2, and 3 affected, we were curious to see, are these the important exons that are represented in the human hypothalamus, which is the region that's important for energy balance? And so when we looked at it, this is just pulled samples from control adults. So looking at the hypothalamus and the hippocampus, you can see that actually, first of all, that there's different patterns of expression depending on the region of the brain, and that the first 3 exons are actually, comprising quite a significant component of the RNA that is expressed from BDNF. And in fact, it's over half, so about 64% of the hypothalamic BDNF includes exons 1 through 3. And so because of this, we then decided to do some mouse studies, and by disrupting exon 1, 2, 4, and 6, which are the 4 exons that are the most commonly expressed in both humans and mice, we hypothesized, just based on our observations that 1 and 2 seem to be important, that that loss of one or two would cause obesity, whereas 4 and 6 wouldn't. And indeed, that's what happened with the mice. You can see exon 1 disruption causes, this is the black line is the mouse that has Bdnf1 knockout, and you can see much higher weight compared to its wild-type littermate. Exon 2 also has significantly higher weight, but you can see for exon 4 and 6, it's pretty much superimposed. The weights are not significantly different. So the mice and the people mirror each other, which is helpful because when you're thinking about studying treatments, that means that you can use the mice as an example. So the other thing we wanted to look at is whether or not pair feeding, so restricting the food to be the same amount as what the wild-type littermate is eating, can control the obesity. And what this is showing here is that if you begin pair feeding, you can see that the mice that were pair fed, which are the gray squares, are identical in weight to their wild-type littermates compared to those who are allowed to eat ad lib as much as they wanted. And this is true for both exon 1 and exon 2. You can see that pair feeding pretty much prevents or does prevent the obesity in the mice. And so the clinical implication is that if we can figure out a treatment for the hyperphagia in people, then we can prevent the obesity in the rare patients who have WAGR syndrome, so the 1 in a million. But perhaps maybe this is an approach that could work if there were common variants in Bdnf as well. And so to look at this, we used post-mortem hypothalamic tissue in 84 control adults who had sudden death. And the region of the ventromedial hypothalamus is here. And what we did is we looked at the expression of Bdnf in relation to variants that are found in the Bdnf locus. And what we observed is that one particular variant called RS12291063 is significantly associated with ventromedial hypothalamic Bdnf expression. And what's interesting is that this is a T to C genetic change that occurs, with not that low of a frequency. So the minor allele frequency is 0.21, and the location of it is right here between exons 8 and 8H. So it's basically an intronic STIP. And what we observed is that this is in the small cohort of adults with cadaveric brain tissue. And you can see that those with the CC genotype had the lowest expression of Bdnf in the ventromedial hypothalamus, and that those same individuals with the low Bdnf had the highest BMI at time of death. So low expression of a protein that tells you to stop eating is associated with high BMI. So that was a post-mortem cohort. So what we wanted to do is confirm this in a larger cohort of healthy children. And so we had over 800 kids that were enrolled in this, and they had a wide range of body mass index from thin to severe obesity. And what we observed is that the CC genotype, which is the variant that is associated with the lowest expression of Bdnf in the brain, was associated with the highest BMI in the kids. And also if we used percent body fat, so looking at body composition, the patients with the CC genotype also had the highest adiposity. Then we confirmed this in larger other cohorts. So in total, over 30,000 individuals. We looked at a Hispanic cohort, a predominantly African-American cohort, another large African-American cohort. And so together, all of these confirmed our findings that the CC genotype is associated with greater adiposity and BMI. So what does this variant do? So when we think about the T to C change from the major to the minor allele, it turns out that you lose a known transcription factor site. So it was previously known that this AAT, AAC, or ACC sequence binds to a protein that was an enhancer element for the DES gene. But what the protein was not known at the time. So we wanted to figure out what that protein was. And so what we did was we did some binding studies to show that the nuclear proteins like to bind to the T sequence, not the C. So you can see it's lighter there. And then if you try to wash out with unlabeled T or C, you can see that again, the binding is preferred to T. You can do a washout there, whereas C is not preferred. So binding is better with T. which is the major allele. Then we did a functional test where we looked at luciferase reporter expression, and the T allele has higher expression compared to the C allele. So that would suggest that decreased binding of some protein to the C allele is associated with decreased expression. So then we used mass spectrometry after protein isolation to identify what this protein was. And it turns out to be heterogeneous nuclear ribonuclear protein D isoform B. And so this is a protein that binds to double-stranded DNA and unwinds it to destabilize this double helix. And this allows the transcriptional machinery to come in and associate with the TATA binding protein complex to initiate transcription. So then what we did was we did siRNA knockdown of this specific protein. And here this is showing that we reduced the protein expression. And after we did that, you can see that the ability of the T allele to enhance luciferase reporter activity is now diminished to the same level as the minor C allele. So by removing this nuclear protein presence, then we kind of lose the ability of the T allele to be an effective enhancer. So this then led us to think that perhaps we could have a precision medicine approach for obesity, thinking about Bdnf insufficiency as a spectrum from the rare to the common. So we know that the minor C allele of RS1229163 is associated with lower BMH Bdnf expression, higher BMI and adiposity, decreased binding of this nuclear protein, and decreased gene expression. So then perhaps we can use Bdnf signaling therapies to be beneficial for weight loss in rare patients who have heterozygous loss of Bdnf, so kind of really full-blown severe obesity. Plus, it could maybe be helpful in common forms of obesity because patients with the CC variant are actually quite prevalent in individuals with African and Hispanic ancestry. So it's roughly about 10% or so. So it's actually, not that rare. So now shifting gears a little bit to think about Bdnf and pain perception. So Bdnf appears to play a role in modulating nociceptive sensory inputs from the dorsal horn of the spinal cord. So when you stub your toe, that signal goes from your toe to your spinal column, and there it has to have a synapse to relay that message to the brain. So it's that that in-between kind of relay that is amplified by Bdnf. So if you don't have enough Bdnf, then the signal to the brain is lower. If you have too much, then you can induce actually hyperalgesia or excess pain for things that are normally not painful. So the Bdnf heterozygous knockout mouse displays decreased thermal pain response. So you can put them on like a hot plate and basically increase the temperature much higher before it like squeaks or pulls its paw away compared to its wild type litter mates. And if you give extra Bdnf into a wild type mouse into its spinal column there, it actually induces thermal hyperalgesia. So it becomes hypersensitive to pain. And then if you give it antisense Bdnf, it actually decreases the pain response. So there have been to this point some anecdotal reports by parents of high pain tolerance in some patients with WAGR syndrome, and that kind of raised the question: is Bdnf involved there as well? So we had families complete a questionnaire that asks about typically painful situations like ear infections, broken bone, pancreatitis, things that are typically painful, and asked, when your child had these types of conditions, did they, have responses to it? And the patients with Bdnf loss scored lower in terms of, having less symptoms of pain compared to those with intact Bdnf. So we want to make sure, since a lot of the kids with WAGR syndrome are receiving vincristine and various other chemotherapy agents if they have Wilms tumor that could, alter nerve function, we conducted nerve conduction studies and observed that everything, is functional in terms of peripheral nerve functioning. And then we also, since the initial studies were done as questionnaires that parents filled out, we also asked the children themselves. So if you hold this little thermode in your hand and it changes temperatures to really cold or really hot temperatures, and you rate on a FACES scale, how uncomfortable it is, you can see that the patients with Bdnf deletion, the yellow bars, individuals have lower feelings of pain related to hot temperatures and cold temperatures. This is more quantitative. You can see over across hot pain ratings and cold pain ratings, that the BDNF deleted individuals have lower scores. And we further confirmed this in rat studies. I'll kind of just go through these quickly, but it's simply saying that the rats, like the mice in the previous reports, basically, if they have BDNF loss, they withdraw their paws at a later point in terms of responding to heat and cold discomfort. And this is showing the latency to response. So it's much later, so it takes longer for them to pull away. And then this is looking at licking of the paw and lifting of the tail in response to hot and cold temperature pain. And you can see that having loss of BDNF basically makes them pain insensitive. And then this is trying to compare sort of A delta versus C fiber, so sort of pressure and heat versus like sharp pain responses. So the A delta responses, which is what this little laser thing is trying to show, is also affected. So basically, the rats have decreased pain response to these painful stimuli. So the next thing we wanted to look at was neurocognitive functioning. So the BDNF heterozygous mice have learning deficits and impaired social interactions. What that means is when they had the mice like swimming in a water tank looking for a platform to rest, the mice with BDNF loss had a harder time learning from trial to trial where that platform was, because it would take them just as long to find it. And for social interactions, it's the ability to recognize another mouse as being novel or new, and how the mouse interacts. So in humans, there had been some published reports of how BDNF may have a role in autism spectrum disorder. So there are altered serum concentrations of BDNF. Various different variants of the BDNF gene have been associated with autism spectrum disorder. And then Rett syndrome, which is caused by MECP2 mutations. MECP2 is a transcriptional regulator of BDNF. And so that kind of sort of ties things together. And in fact, even common variants of BDNF have been associated with poor cognitive performance on various different map routing tasks and things like that. They're considered like executive function tests. And then there was a paper that Kelly Trout and Catherine Lewis actually were co-authors on this paper that showed that in a case series of 54 patients with WAGR syndrome, that there was about a quarter of them had been diagnosed with autism spectrum disorder, and about 3/4 have been diagnosed with intellectual disability. So we wanted to look at this in relation to BDNF, and observed that in those with BDNF deletion, that there was lower cognitive functioning, as well as lower adaptive behavior. So that includes performance on an IQ test, as well as the Vineland scales of kind of daily living and being able to be independent. So you can see here the significant decrease there and there. One thing I should point out is that because all of the patients with BDNF loss have larger deletion sizes, that could be a confounding factor. So what we did was we looked at those with and without BDNF deletion to see whether or not deletion size affected things. And it actually didn't correlate with IQ. So it seems to suggest that it's not really how big the deletion is, but it's whether or not the deletion actually encompasses the BDNF spot. So it's just like, it just keeps going, and then when you actually have BDNF involved, that's when you see kind of the bigger drop. So it's about a 20-point drop that you can see in the IQ there. Okay, so the other thing that we looked at was autism spectrum disorder. So this can be done through an interview kind of mechanism, as well as observed testing by a neuropsychologist. So when you look at it based on the interview, you can see that social interactions are significantly impaired in those with BDNF deletion. And that because of that, the overall scores are such that a higher percentage of people meet criteria for ASD. The one thing, though, I do want to point out, though, is that when you have actual clinical neuropsychologists doing the testing, that full-blown autism, like really big, rigorously diagnosed autism using rigorous criteria and clinical judgment actually is not significantly different. And so I think that one has to be cautious, because ADIR, the interview process, may overdiagnose like the ASD diagnosis because there may be confounding factors with cognitive and visual impairment. Nonetheless, I still think that if a child carries the diagnosis, they can receive very helpful interventions for their cognitive development. So I don't think there's, we don't need to like be so worried about the labeling, but just really understanding that it's a different kind of autism or a different flavor of autism spectrum that we're talking about here. Because really, when you use a rigorous ADOS criteria, really full-blown autism is not necessarily higher in those of Bdnf deletion. Okay, so I think we've really talked about Bdnf quite a bit now. So we'll move on to PAX6 and some of the other genes. PAX6, we already know is related to eye development, but it's also important in brain and pancreas development. And so what we did was we wanted to look at whether or not PAX6 could be associated with some, other, features. So these are brain MRIs. Here's a healthy control that shows a normal pineal gland. And then here's a person with PAX6 loss. So this is people with WAGR syndrome and PAX6, or also people with point mutations of PAX6, which is isolated aniridia. So these individuals all, you can see now, like you really don't even see the thing here. It's like, it's so small, you can't see it or it's missing. So there was pineal hypoplasia, and the pineal gland is important for making melatonin. So melatonin is a sleep and circadian rhythm hormone. So we noticed that there was lower serum melatonin as well as a metabolite for melatonin in the urine. And this correlated with greater sleep disturbances. So, this could have clinical implications in terms of thinking about how PAX6 could be involved in both melatonin secretion and sleep. There's another gene in that region called C11OR46. And this gene is believed to have a role in chromatin remodeling. And so when it's combined with PAX6, it seems to cause even further problems. So we've observed, not just us, but us and previous researchers have found that PAX6 loss is associated with a smaller corpus callosum, that's the connector on the two halves of the brain, and that if you also have, what we observed is that if you also have C11OR46 involved, you get an even smaller corpus callosum. And in particular, it's the posterior part that's affected. Probably a picture is a little bit better to show this rather than these little bar charts. Okay, so what you're seeing here is this is a child and an adult who are control healthy individuals, and the CC stands for the corpus callosum. And you can see if you go to PAX6, there's child and adult, you can see that it's smaller, and then it's even smaller when you get to PAX6 plus C11OR46. So that shows us that this is an additional important gene. And then we're trying to now explore even, other regions. And so this is using brain morphometric mapping. And so that's really looking at the surface and the three-dimensional structure of the brain. And so what this is telling you is that the blue are regions with increased morphometric similarity compared to controls, and red is reduced. And so what all this is showing us is that we can relate a genetic finding with an actual kind of brain difference. And then what researchers are doing now, and this is in collaboration with other researchers, so my group is collaborating with others who are looking at this, to see whether or not there's specific cells within the cell types within the brain, certain gene expression, or other things that are being affected to kind of explain these. And this can all lead to potentially future targets for treatment. So in terms of thinking about how the corpus callosum, because of the left and right brain connections, might be affected, one of the things that comes to mind is auditory processing. So when you don't have appropriate left-right connection, then you can actually have these defects that are not necessarily related to hearing per se. So hearing loss is similar. So people with WAGR syndrome and aniridia don't have worse actual hearing, but in terms of and this is detected through like brainstem responses and hearing tests. However, when you look at auditory processing, and what that is say, for example, like if somebody like is talking on one side and the other side, somebody else is talking, and you're trying to like hear both conversations at the same time, or you're just in a noisy environment and you're trying to like hear the one conversation that's important. It's more being able to filter out and hear specific things. That's what's affected. And so, what we've observed is that patients with WAGR syndrome have a much sort of a twofold higher problem with auditory processing compared even to patients with aniridia. So I think that, all of those together, point to the importance of how having these morphologic changes of the brain can affect function. Let's see, a few other findings. So we found that the larger the deletion size, the shorter the stature. So for every an additional 1 million base pairs that's deleted, there tends to be about 1 centimeter shorter adult height. So that would suggest that there's probably multiple genes along chromosome 11 that are, affecting growth. Also, there seems to be some craniofacial differences. So some narrow, high-arched palate, some fibrotic bridging in the palate is shown here, and then there's just like a difference of positioning. So there's upper pharyngeal constriction, a shorter anterior cranial base, lip protrusion, and then the hyoid bone. And so the hyoid is the one in the neck. So that this can affect, if someone has obesity and these things, you can see how this could exacerbate things like obstructive sleep apnea and other kind of breathing concerns. Also, WT1 is important in Müllerian regression. So sort of the default state during embryogenesis is female, and so we have to have genes turn on if we want the embryo to develop into a boy. So what happens is everything sort of default starts off as girl, and then you've got like genetic signals to tell you to not to sort of regress the uterus and all the other female structures, the Müllerian structures. And so what we've observed is in patients with WAGR syndrome who have WT1 deleted, this is an example of a boy who had recurrent urinary tract infections over and over again. And it turned out when we did an MRI, what we found was there was this pouch inside of the pelvis that was contiguous with the bladder and it was filled with all this fluid and debris. And so it turned out that this was a leftover sort of uterine remnant in the boy that was basically collecting urine, and all that sludge and all that stuff that was in there was actually, causing the recurrent urinary tract infections. And so we measured anti-Müllerian hormone. It was undetectable in this boy. And this arrow is showing you that he indeed is a boy. He has a normal phallus appearance, but he has this uterus-like structure as well. So I think these are important things to be aware of that, are involved in the spectrum of the disorder. Okay, so now I'm going to shift gears a little bit and kind of talk about, this partnership of researchers and families working together. So in the process of developing that very complex schedule, it was really important for us to receive feedback from families because really it was a very challenging schedule. And so we tried our best to figure out the feasibility based on this feedback for the proposed evaluations, but then also modified as we went along. And then families also brought attention to us the things that needed to be looked at because parents know their kids the best. And so by hearing what families had to say, that's what drove us to know what we needed to study. So balancing research with clinical care was also the thing we were trying to integrate into this because there were some clinical concerns that we wanted to address at the same time as doing investigations to look for and to uncover other potential new problems that were undetected. We also had to really modify what we were doing because children do have special needs. And we tried to do a lot of the testing in the morning for the things that require concentration, trying to make things kind of consolidated in the same location so people didn't have to walk back and forth. Taking frequent rest breaks was really important. And we had to adapt our testing for visual and intellectual impairment. So this is an example of our IQ testing. Instead of having people look at shapes, we had them feel shapes. And so these are things that were important to allow, for testing across the spectrum of abilities. So this is kind of the timeline. So in 2002 was the first time I ever met anybody with WAGR syndrome. Up to that point, in 2002, I actually hadn't even met anyone with WAGR syndrome yet. That was when I was learning about WAGR syndrome for the first time during my pediatric residency training, but I never had met anyone. And it was only 2 years later that I started to become interested in endocrinology and genetics of obesity. And then in 2006 was when I Googled for the WAGR syndrome and actually found the International WAGR Syndrome Association. And that's when I met my first two families. So basically 4 years after first hearing about WAGR syndrome was when I finally met somebody with WAGR syndrome. And so the first two people that I met with WAGR syndrome are Caroline and Irma, and that's a very precious zebra that now my daughter loves. So it's a very just an exciting and wonderful thing to have this long-term relationship with the families that I've met during the course of my career. And then in 2007, I met with everybody at WAGR syndrome, and it was there that I was able to start getting some research studies done by collecting blood samples and mailing in records. In 2008 was when the on-site study at the NIH happened. And then in 2014, I moved to Memphis, Tennessee, and I had to stop doing the on-site study at the NIH, but I continued to analyze data and samples from the time that I was at the NIH. So 6 years later, I'm still continuing to collect samples and data and continue to do analyses. For example, in collaboration with Dr. Larry Ryder, we've been collecting primary teeth when they fall out to use the dental pulp stem cells to grow neurons, nerve cells. And then we can also use blood to isolate white blood cells and then turn them into pluripotent stem cells. These are kind of the way to not use embryologic cells, but rather cells in children and adults to be able to create other different types of cell types. And one of the things that we're working on is trying to really make these neuron types to be like the brain ones, the hypothalamic neurons, so that we can test out various different drugs for treating Bdnf insufficiency. So some of the things that we learned about patient recruitment and retention is that you can get a lot done through the mail, which is great. We met a lot of people, at WAGR weekend, but we also were able to contact people internationally who could mail samples to us. And some of those we were very fortunate to be able to meet in person. And one of the things that was really great was that patients who came to the NIH blogged about their experiences on the IWSA website, and I think that helped other future patients to be more comfortable because they kind of knew what to expect. People kept journals and even passed a quilt from one patient to another, and that kind of made sort of the family feel of this whole experience, much more close-knit community. And then our team then would go to WAGR weekends to share our results, and then it kind of really closes the loop in terms of the researcher and the participants, and going back to the participants to share what we learned. So some things we modified because of feedback was some of our testing for the pain testing, sometimes caused some discomfort for people because we sort of realized that we weren't able to really give proper instructions so that people could understand sometimes what we were really asking. And so we were kind of putting people through really extensive testing without really getting useful information from it. So that's kind of why we eliminated some of the really lengthy tests that really didn't yield pertinent information. And then we were doing meal testing with a lunch buffet array that was lots and lots of different foods, but for someone who's visually impaired, you can't really access all of those things. And so we modified things to being a single food item so that it's easy to pick up with your hands, and a chicken nugget. So it's easier and much more replicable. Also, we observed some adverse reactions too. So we found out that propofol, which is often used for sedation, it's a drug that's carried in a soybean oil. And so in people who have baseline hypertriglyceridemia, it can be a risk factor. And so now we would advise before getting propofol that people check to make sure that there are no baseline risks, to receiving anesthesia, especially, if there's high triglycerides. We also involved rehabilitation medicine for our patients because we wanted to have an occupational therapist with low vision expertise to give advice as to how to make adaptations for better independence. And audiology was very helpful in terms of giving feedback about how to help with auditory processing. This is an example of a chart that the low vision specialist showed me. It's a Paley-Robinson test that kind of, you can see that the contrast, most individuals can read all the way down to here, whereas people with WAGR syndrome oftentimes can't even distinguish things, once you get past the first two lines. So if that's the case, if this is what the whole sheet looks like to someone with WAGR syndrome, then, it's really important then that you select proper writing paper for people to use. So you want something with really good contrast, because here, if you're handing a child in school a light blue paper with very faint white lines that you can barely see, of course no one's going to write on the line. They're going to write in any direction they want. But if you give them a piece of paper that looks like this, then they can see it and then they can write more neatly. So like a lot of families were saying the kids were writing erratically all over the page. Well, it's because they can't see the lines, and I think realizing that can make a big difference in terms of school performance. Also, one of the things that can be helpful is yellow tinted glasses can actually help with some of the contrast and reducing glare, is what the occupational therapist shared, and a lot of families gave feedback that it did work. Other things was in terms of tying shoelaces, instead of trying to deal with the bunny ears kind of thing, was to use these coily spring-like shoe ties. So you just pull and let go, and it basically allows you to tighten your shoe. And so for some kids who have been using Velcro shoes only because they had trouble with tying, this was their chance to wear regular shoes, and it sort of expands their choice of options to use. And in terms of feeding yourself and being independent, having a plate that has this sort of edge to it and a gripping spoon that has, that goes around your hand and is bent like this makes it so much easier for you to have independence. And so these are all the kinds of things that the occupational therapist supplied. For the audiology component, using an FM device was advised, because by having a device in your ear that magnifies what the teacher is saying allows you to hear that one louder than all of the background noise of the other kids. And also being in the front of the room helps too, so you don't get distracted by background noise, because the problem with auditory processing is not physically hearing it, but it's actually filtering out things you don't want to hear. So it's being able to selectively hear what you want to hear. So by really amplifying the signal to noise ratio, then you can improve the signal. So over time, we were able to present our findings as abstracts at scientific conferences, publishing in medical journals, summaries in the IWSA newsletter and website, and presenting at WAGR weekend. So these are the really important way of getting the word out about what we're learning, because even though this is a rare condition, this is so important and valuable information that we want the whole scientific community and medical community to know, and we also want to make sure all the families know as well. So there were some few things that we learned from these experiences. So the family perspective is really important, to have a novel angle to examine things. So there's some things that even among my patient populations that are non-WAGR examples. So risks for childhood obesity, for example. So there are major barriers that are preventing children from economically disadvantaged areas from engaging in outdoor play. And so, one of the things that everyone assumed was that there was, a concern for safety and that there was gun violence. And while this was important to a lot of families, if we hadn't asked an open-ended question, we would have assumed that was the only problem. Like, we would have never even realized that there was yet another thing that was really important. And yet, if we hadn't asked, we wouldn't have known. And guess what it was? Stray dogs. Like, it was basically something that was not even, on people's radar. And it was because, in these research studies, people actually asked people, open-ended questions about why, that we learned that it was, by simply, having more animal safety and dog catchers to get rid of stray dogs, that it made playgrounds safer. So it was not just gun violence that was the issue, it was also stray dogs. And so if we didn't ask, we would have never known. So I think that this example can be extended to what I learned in WAGR syndrome too. So several patients were being described as having some clumsiness because they were bumping into objects kind of, at their periphery as they were walking by things. They would knock over things. And so a lot of people are just assuming, well, it's just probably visual impairments or maybe neurocognitive deficits that are doing this. But then, we started kind of getting more information. So some patients were reporting double vision whenever they turned their, whenever they looked at things. So they had to like turn their head, otherwise they would end up with this double vision. And so they would, kind of move like an owl, I guess, and when you hear these descriptions, then you think, wait, this is something a little different. This is not just run of the mill, not seeing things. And so when we ask patients to look, so this boy is being asked to look to this side, you can see that this eye is, turning to the side like it should, but this one's getting stuck in the middle. And so you can imagine if the eyes are not lined up, then you're going to see double vision. But if you also can't get your eye to go to the side, then of course you're going to have problems seeing things on the side, on your periphery. You're going to knock over things naturally. So we then did an MRI to look to see, was there an explanation for this? Well, it turns out in mice, there had been some reports that PAX6 is involved in the abducens nerve. Abducens means the nerve that abducts or draws the eye to the side. And so it turns out that we actually have this, missing. So this is the normal one, and this is the missing one, in almost a third of patients with PAX6 and or WAGR syndrome. So this is a really important new finding. Sorry, this is just showing you the normal and the absent. Okay. So the other thing we learned is sometimes the priorities of researchers and families may not always align. And so when that happens, we have to really, think about this, because what's best for science may not be the best for patients. And so I think I alluded to this earlier about autism diagnosis. So being really precise about the diagnosis of autism features, is important for research, right? But we have to think about, do we need to be so precise that we start, kind of affecting diagnoses? Because what happens is having social services oftentimes is possible if you have a label of autism. So, one thing may be that in research, we're going to like go through, with like a fine tooth comb to like really tease out, is this true autism, or is this just like a flavor of autism? But when it comes to clinical care, I think we should, maybe consider, kind of being a little more generous or broad in our definitions, because having the diagnosis of autism can gain really beneficial things for the care of patients. So then I think it's important to communicate with families about the semantics of diagnostic criteria, separating clinical and research charting so that it's clear what we're talking about, and not put at risk some of the services that patients can benefit from. Also, there's one other thing that we kind of experienced that alerted us to how important it is when we think about rare disorders, because sometimes there may be different patient organizations that are trying to compete for the same small group of patients, and this can lead to some schisms and problems. And so we actually had one case where an international patient was enrolled in our study, and they'd given us records, prepared all the pre-visit paperwork, got the visa to travel, landed on US soil, but then never came and never contacted us ever again. And we later found out that they were rerouted to a different city for an aniridia-specific research study by a separate organization. And, that whole experience, I think, saddened me, because I really would love for all researchers to collaborate, because I think that's how we benefit patients the most. And so I think I just wanted to mention this, just, because it is something we learned and something we experienced, just so that people are aware that this does exist. Because truly collaborative science is the best, and that's, what we really should be aiming for. So overall, we've learned, that researchers, really, have, just like limits in terms of areas of expertise. And so that's why collaboration is so important, because WAGR syndrome, for example, affects essentially every organ system. And so every one disease process requires understanding another. So for example, vincristine, for Wilms tumor could exacerbate nociception deficits, so we need to look at that. And that's why having a neurologist on board was really important. Obesity can increase the risk of pancreatitis and renal disease. So like really having nephrologists and then endocrinologists, and the GI specialists on board were really important. Auditory processing, visual impairments, all of those can contribute to developmental delays. So having the audiologist and the ophthalmologist involved with the behavioral psychologists were also very important. So that's why having like all these puzzle pieces together for collaborative science is what's best. And that is, what I would really encourage all researchers to shoot for, is to be collaborative. And so on the note of collaboration, thanks to all the people who've made this work possible and our funding agencies as well. So thank you very much.